All right. I'd like to thank the JGI for the opportunity to present my work today. I'm John Chodkowski. I'm a senior graduate student in Ashley Shade's lab at Michigan State University. And so ecologists realize there are many links between the environment, the physiological state, and the microbial community that lead to microbial processes. But what's missing from this picture is microbial interactions and the interplay of these microbial interactions between the physiological state and the microbial community, which lead to these microbial processes. And so there are many ways in which microbes can interact, and the focus of my project is specifically on the subset of these interactions that are mediated through exometabolite production. And so with focus on one cell, exometabolites are pretty much all the extracellular chemicals that are secreted by one cell that can affect surrounding neighbors. And so these can include things such as antibiotics, signaling molecules, and even central metabolites as well. And so there are many different forms of these interactions that can take place that range from cooperation to antagonism. And it's really the combination of all these interactions that can have ecological consequences. But one can imagine it's pretty hard to study these interactions in nature. And so as an alternative, we and other researchers have started to propose the use of synthetic communities. And so simply speaking, synthetic communities are laboratory scale systems that try to study microbial communities either using top-down or bottom-up approaches. And you can see that we can control a lot of parameters in the system. And an advantage of this is that we can also start to quantify some of the microbial interactions through the use of mass spectrometry. Another aspect about this is that many wild microbes, at least speaking for bulk soils, do live in this nutrient-deprived state. So using this synthetic community, we can try to study these microbes in a physiologically relevant state. And so the best way we can mimic this state is to try to study these microbial communities in stationary phase. And even though stationary phase, there is no growth, we know that there's constant protein production and activity as well. Another aspect about this is that exometabolite production is linked to nutrient stress or stationary phase, as seen here. So here I'm showing a cladogram and other biotic drivers as well. And so here I'm showing a cladogram of different microbial species, and each microbial species is color coordinated to the biotic driver that leads to the production of a particular exometabolite. So putting this all together, we can use synthetic communities to study microbial interactions under physiologically relevant states. And so what I'm showing you here is a synthetic community that we use in our lab, which I'll refer to as a transwell system. This is composed of two parts, a filter well plate and a shared media reservoir. And so in the 96 filter well plate, each filter is equipped with a 0.2 micron filter bottom. So what this allows is for the microbial community members to be physically separated, but to interact chemically through the exchange of exometabolites and to share community resources in the shared media reservoir. And so we can use this synthetic community system to answer a couple of questions, broad questions. What population level and specifically transcriptomic responses result from interspecies exometabolite-mediated interactions? And what are the community level signatures characteristic of those interactions? So today, I'll mostly be able to get to question one with a little snippet of question two. So first, I'd like to also thank the JGI. We were awarded a community science program, and they have helped us generating an immense amount of data. And so here are the three members that we use in our synthetic community. We have Burkholderia, Chromobacterium, and Pseudomonas. These environmental type strains were chosen for a few reasons. One, we have their genomes and they're pretty well annotated to make sense of the transcriptomics. Two, we have reported from the literature and in our lab that these microbes have the ability to interact. And three, what's of interest to us are these biosynthetic gene clusters. So these are characterized exometabolites in for each of these community members that we can use to try to interpret the mass spec data. And so when I refer to biosynthetic gene clusters, I'm referring to a subset of exometabolites that I'll refer to as secondary metabolites that are used specifically for competitive interactions. So for our experimental design, we have a replicated time series. We have all of these community compositions. We have 6 time points, the first time point being at exponential phase, and then 5 time points into stationary phase, where we have 4 biological replicates for each time point. And then 1 plate ends up being 1 experimental unit where we can measure population and community outcomes. And what I mean by this experimental unit, I'll show in this next slide. So to measure population outcomes, there are certain wells assigned for measuring population outcomes for each member, as seen here. And then the community outcomes can be taken by obtaining the spent medium from the shared media reservoir and performing mass spec analysis on that. So again, I'll only have most of my time to present some of the transcriptomic work. So let's dive into the data. So what we see here is a Pca analysis. So the color is, so specifically we're looking only at the gene expression of Burkholderia. And so the colors correspond to the community composition, whether Burkholderia was in a full community, in cold culture with Chromobacterium, in cold culture with Pseudomonas, or in monoculture, whereas the size of the points represent the time series. And so each point, in essence, represents a summary of the gene expression response of Burkholderia in that particular community composition at that particular time. And so points closer together in space are more similar in the transcriptional response, while points further are more dissimilar. And so some things that we see is that we have this replicate reproducibility by the tight clustering of particular time points. We see this growth phase differentiation between the exponential phase and the stationary phase time points. And third, we see this general temporal trajectory in each of the community compositions. And so what's more striking as we look at the other PCAs is that neighbors matter in terms of driving transcriptional response. So what I'm showing here is the PCAs of the other 2 members in the community, Pseudomonas on the left and Chromobacterium on the right. And so what this divide and what I'm highlighting here is the fact that the transcriptional response seems to be driven by the presence of Burkholderia. And even when we build this up to a full community, when the other member would be present, it doesn't matter because we see this high overlap in terms of Burkholderia driving the gene expression. And so when we do variance explain, we see most of it's explained by the community composition, while time explains that to a lesser extent. So we next want to ask how, or how Burkholderia is sort of driving this transcriptional response of its neighbors, and we hypothesized that it was competitive interactions mediated through secondary metabolite production. And so for that, we took a network approach. So here in green, let's just say that's Burkholderia gene expression, and in blue, let's say that's either Pseudomonas or Chromobacterium gene expression. And so what we did is we used the transcriptional data to run a co-expression network, and then specifically subset the nodes that had interspecies connections. And then we ran module detection and functional enrichment. And so to step back first, I would just want to point out that just looking at the transcriptional data, of the 20 biosynthetic gene clusters in Burkholderia, we saw 5 of these that were upregulated when comparing the cold culture conditions to the monoculture, suggesting that Burkholderia was upregulating these secondary metabolites and becoming more competitive. When we looked at that, when we looked at the interspecies network, in this interspecies region, we were able to capture 4 of those secondary metabolites in an interspecies region, suggesting that secondary metabolites from Burkholderia had coordinated gene expression toward its neighbors and potentially driving these transcriptional response. And one that I would like to highlight here is this molecule called capsaicin. And so this was a molecule that was previously characterized to have reported antimicrobial activity by binding to the RNA polymerase. So it does suggest and put forth this idea that it is potentially the competitiveness of Burkholderia that's driving the gene expression. Here I'd like to point out that this is one gene involved in the gene cluster of capsaicin, and specifically looking at the Burkholderia gene counts in each of the 4 community compositions, the full community in cold culture with Chromobacterium, in cold culture with Pseudomonas, and in monoculture. So what we notice is that this is not expressed at all when alone, but when with neighbors, this starts to be upregulated. And what's interesting to us as well is this non-additive effect. So if you were to assume the naive assumption that in the full community, it would simply be the summation of the two pairwise. We see even an increased amount of transcription of this as well. So it almost seems like in the full community is becoming even more competitive. Additionally, we were able to identify this molecule using mass spec, and so we know it's there. And then specifically looking at the abundance of that through time, we do see a sort of pattern in terms of the transcriptional response of increasing through time and also the abundance of this secondary metabolite increasing through time as well. So in summary, with this question, it seems that both composition and time contribute to gene expression dynamics. Burkholderia seems to drive the transcriptional response of its neighbors. And when trying to figure out how Burkholderia is driving the transcriptional response of its neighbors, it seems that the upregulation of secondary metabolites and secondary metabolites involved in coordinated interspecies gene expression point to exametabolite-mediated competition. And so this is ongoing work. So I'll give you a little snippet of what we're also trying to do. So this is characterized exametabolites provided by the JGI. And so what you're seeing here on each column is a different community composition, all 7 community compositions. And underneath each of these community compositions is the temporal progression of that exametabolite. So what we're trying to do is, well, what you notice first off is just the compositional and temporal trends in the data. But with this, what we're trying to do is link some of the transcriptional responses to the exametabolite profiles, and also start asking if there are particular community exametabolite outcomes that are characteristic of certain community compositions. So with that, I would like to thank the Shade Lab and the JGI again.
Thank you very much. We have a time for one question.
So,
do you have any examples of where a precursor that was excreted from one is then used to make a specialized metabolite from another species, by another species?
So with that exametabolite data that I showed you, they are characterized. And I haven't directly linked it yet, but using KEGG pathways, I can give you one example. We find that sulfate is being taken up by the medium from Burkholderia. In combination with a serine-derived metabolite, gets used to make cysteine, which also cysteine is directly incorporated into a siderophore molecule as well. So following the KEGG pathways like that, we do see that, but I haven't made the direct link yet.